placed. This is achieved by the second scent will rise up in the paper. After a few minutes, we can see. Hello students, welcome back. From a young age, we learned that plants make their own food, particularly carbohydrates, through photosynthesis. Since they produce their own food, these organisms are categorized as autotrophs. All other organisms that depend on the green plants for their food are known as heterotrophs. If someone asks you what is the significance of photosynthesis, this should be your answer. Photosynthesis is important for two reasons. First, it is the primary source of food on earth. Second, through photosynthesis, plants release oxygen into the atmosphere. Without oxygen, animals cannot survive on earth. In lower grades, you might have conducted experiments showing that photosynthesis needs carbon dioxide, chlorophyll and light. Similarly, in earlier times, scientists performed a number of experiments which poured light in understanding photosynthesis. In 1770, Joseph Priestley conducted experiments and discovered that air is vital for the growth of green plants. In 1774, he made the groundbreaking discovery of oxygen. He placed a mouse and a burning candle in a bell jar. Eventually, the candle went out and the mouse suffocated. Today, we know that a burning candle and a mouse respiration both release carbon dioxide. Carbon dioxide reduces the surrounding oxygen, causing the mouse to suffocate and the candle to go out. In 1770, Priestley conducted experiments that revealed how both a burning candle and an animal's respiration deplete the air's quality. He placed a mint plant in the jar and noticed that the candle kept burning and the mouse lived. He concluded whatever substance the animal and candle removed from the air was replaced by the mint plant. We now understand that mint leaves release oxygen into their environment, which explains why the candle continues to burn and why the mouse survives. Later, Jan Ingenhousz used the same experimental setup and placed one jar in sunlight and the other one jar in dark. Despite the presence of a green plant, the candle went out and the mouse got suffocated in the jar which was placed in the dark. Thus, he showed that sunlight is essential to the plants to release oxygen that purifies the air. His experiments with aquatic plants revealed that small bubbles formed near the green parts of the plant in bright sunlight, but the bubbles were not formed in darkness. He later identified that the bubbles were of oxygen and he concluded that only the green parts of plant can release oxygen. In 1854, Julius Van Sachs proved that plants produce glucose while they grow. He demonstrated that glucose is produced in the green parts of the plants, which are found in special structures. Now we know that these green structures are chlorophyll and glucose produced stored as starch. Later, W Engelmann performed one experiment to show the first action spectrum of photosynthesis. What is action spectrum? Action spectrum means the graph of the rate of photosynthesis and which all colors of light are best for photosynthesis. He took some green algae named cladophora in the suspension of aerobic bacteria. The significance of aerobic bacteria is that they divide rapidly when oxygen is available. Aerobic bacteria will divide rapidly when oxygen is available. Using a prism, he split the light into its 7 components and illuminated the cladophora with this split light. He observed that the aerobic bacteria accumulated mainly in the region of blue and red light of the split spectrum. That means more oxygen is produced in the blue and red area because aerobic bacteria needs oxygen to multiply. Therefore, he concluded that the rate of photosynthesis is higher when the plant absorbs blue and red light. By the mid of 19th century, scientists understood that the plants use light energy to make carbohydrates from carbon dioxide and water. The empirical equation of photosynthesis understood by then was carbon dioxide plus water in the presence of light gives carbohydrates and oxygen. In this equation, CH2O symbolizes the carbohydrate produced. Then came a microbiologist named Cornelius van Niel, who used purple and green sulfur bacteria for his experiment. There is a speciality for green sulfur bacteria that they are photosynthetic. They can perform photosynthesis. He provided light, H2S and carbon dioxide to these green sulfur bacteria to perform photosynthesis. These bacteria performed photosynthesis and demonstrated that photosynthesis is a light-dependent reaction in which hydrogen from an oxidizable compound reduced carbon dioxide into carbohydrates. This hydrogen from this oxidizable compound reduced carbon dioxide into carbohydrates. Then raises the question, is oxygen released during photosynthesis derived from carbon dioxide or water? When Cornelius allowed sulfur bacteria to perform photosynthesis in the presence of H2S, the final product formed was a sulfur or sulfate depending on the organism. Oxygen was not released. Instead of oxygen, the product formed were sulfur or sulfate depending on the organism. Hence, he inferred that oxygen released by the green plants evolved from water and not from carbon dioxide, because he provided carbon dioxide to the sulfur bacteria but oxygen was not released. Later, this was proved by radioisotopic technique. Hence, water is the oxygen donor for green plants. The correct equation of photosynthesis is 6 carbon dioxide plus 12 H2O in the presence of light gives glucose, 6 H2O and 6 carbon dioxide. C6H12O6 is the glucose formed. We know that the photosynthesis takes place in the chloroplast of the mesophyll cells. The chloroplasts usually align themselves along the walls of the mesophyll cells. This is a mesophyll cell and these are the chloroplast. And this arrangement maximizes the amount of the incident light. Mesophyll cells have adaptations to get maximum amount of light, like they have thin walls and have large surface area. During morning and evening, the sunlight is oblique and there is low light intensity. So the chloroplasts align themselves parallel to the incident light for maximum light absorption. But during midday, the sunlight is direct and there is high light intensity. So the chloroplast align perpendicular to the incident light to reduce the excessive light absorption and reduce photo damage. Now let's learn the structure of chloroplast. Covering the chloroplast, there is a double membrane, an outer membrane and an inner membrane. And there is an intermembrane space between the outer and inner membrane. There are closed disc-like structures called the thylakoids. And these thylakoids are arranged in stacks known as granum. One stack is known as granum and altogether they are known as grana. The fluid filled inside is known as stroma. Grana are connected by stromal lamellae. Stromal lamellae are extensions that run from one granum to the neighboring granum through the stroma. The membrane system is responsible for trapping light energy and synthesis of ATP and NADPH. In stroma, the enzymatic reactions synthesize sugar and in turn gets converted into starch. The first set of reactions in photosynthesis is called light reaction. As the name indicates, this reaction needs light energy to produce ATP and NADPH. The next set is not light dependent, but it depends on the product of light reactions. Which are the product of light reaction? That is the ATP and NADPH to produce glucose. Hence, the second set of reactions is called dark reaction. We will learn in detail about the light and dark reactions in this video. If we closely observe a green plant, we can observe different shades of green in their leaves. Have you ever wondered why? Let's try to find an answer for this through the separation of pigments in green leaf by paper chromatography. You might be familiar with chromatography. It is nothing but a laboratory technique to separate the components of a mixture. A chromatographic separation of the pigments shows that the different shades of green color in green leaves are due to different pigments. Through chromatographic technique, scientists proved that the green color we see in plants is not due to a single pigment. 4 pigments are responsible for the green color: chlorophyll A, chlorophyll B, xanthophyll and carotenoids. Chlorophyll A, blue-green in chromatogram. We can see chlorophyll A pigment as blue-green in chromatogram, chlorophyll B as yellow-green, xanthophyll as yellow and carotenoids yellow to orange. These are the colors of pigments seen in chromatography. Now let's explore the process of chromatography in detail. Take a thin strip of paper and mark a line around 2 cm from the bottom. Now take some green leaves and crush them so that we get a concentrated mixture. Now take a couple of drops from this mixture and place it on the line we marked. Now take a beaker and add a solvent. The solvent used is mainly a mixture of petroleum ether, acetone and water. Dip the paper into the solvent. Make sure that the line on the paper is above the surface of the solvent. Now the solvent will rise up in the paper. After a few minutes, we can see that various pigments are positioned at different distances on the paper. The pigments will vary in color. Here you can see all the 4 pigments: chlorophyll B, chlorophyll A, xanthophyll and carotenoids. Now that we understand how to separate the pigments, let's learn what are the roles performed by these pigments in photosynthesis. The wavelength of visible light normally ranges from 400 to 700 nanometer. We know that there are 7 colors in VIB here, that is the visible light. Various pigments absorb light at distant wavelengths. Let's analyze this graph for easy understanding. Y-axis is the absorption of light by pigment and x-axis is the wavelength in nanometer. When you observe here, you can see that the chlorophyll A, this blue line is chlorophyll A, and this can absorb maximum blue light between 400 to 500 nanometer and some red light between 650 to 700 nanometer. Chlorophyll B, this green line is chlorophyll B, absorbs somewhat blue light and some red light, but at slightly different wavelengths. While carotenoids absorb blue light, this orange one, they absorb blue light, violet and green light. Here is the green light after 500 nanometer. Let's create a graph to illustrate the rate of photosynthesis. In this graph also, the y-axis is absorption of light by the pigments and x-axis is wavelength. And in this graph, we can clearly see that maximum photosynthesis happens at 400 and between the 650 and 700 nanometer. Which pigment is more in this region? Yes, it is chlorophyll A. Maximum photosynthesis happens at blue and red regions. The pigment which is more in this area is chlorophyll A. Hence, we can conclude that chlorophyll A is the chief pigment associated with photosynthesis. From the graph, we can conclude one more thing. Photosynthesis also takes place in the regions of visible spectrum other than blue and red, but of course in a lower rate. That means other pigments like chlorophyll B, carotenoids and xanthophyll are also responsible for photosynthesis. These pigments absorb light at different wavelengths which chlorophyll A cannot. And we know that light is energy, right? These pigments transfer the energy they absorb to chlorophyll A to be utilized for photosynthesis, and the other pigments protect chlorophyll A from photooxidation. Photooxidation means light energy causes oxidation of a molecule resulting in its damage. So they protect chlorophyll A from damage caused by intense light. Photosynthesis happens in two steps: light reaction and Calvin cycle. The dark reaction is also called Calvin cycle. There are mainly 4 steps in light reaction: light absorption, water splitting, oxygen release, and production of ATP and NADPH. In the thylakoids, the light pigments are organized into complexes called LHC, light harvesting complex. This is a light harvesting complex. Here the pigments are organized. They are also called antennae. These LHCs are present in 2 photosystems, photosystem 1 and photosystem 2. These photosystems are named after the order of their discovery, not the order in which they are participating in light reaction. Here, first one to participate is PS2 and then comes PS1. All pigments, that is chlorophyll B, xanthophyll and carotenoids, are present in LHCs, but one molecule of chlorophyll A forms the reaction center. The chlorophyll A molecule in PS2 accepts light at 680 nanometer and hence named P680. Similarly, in PS1, the chlorophyll A forms the reaction center which can absorb maximum light at 700 nanometer and hence named P700. Now let's see how this electron transport happens. In photosystem 2, chlorophyll A forms the reaction center that absorbs light energy at a wavelength of 680 nanometer. This causes the electrons in these pigments to become excited as they gain energy. The electrons that gain energy enter an orbit farthest from the atom's nucleus. Hence, it will be easier for an electron acceptor to capture these electrons. The electrons picked up by the electron acceptor are then passed on to the electron transport system consisting of cytochromes. These are the cytochromes. Cytochromes are proteins that can absorb and donate electrons, facilitating electron transport. Electrons move from a high to low redox potential, like going a downhill. The top of the hill are the oxidizing agents that accepts electrons, while the bottom are reducing agents that donate electrons. The electrons move in terms of redox potential scale. Now they are passed on to PS1. The electrons that are accepted by the electron acceptor from PS2 are now passed on to PS1. The reaction center, which is known as chlorophyll A in photosystem 1, absorbs red light at a wavelength of 700 nanometer, resulting excitation of its electrons. These electrons are picked up by a different electron acceptor, and then these electrons again travel down the hill to a molecule of NADP+, and the electrons reduce NADP+ into NADPH. This electron transport is in the shape of Z, and that is why this is known as Z scheme. So when light falls on P680, electrons gain energy and they get excited, and these electrons are picked up by a primary acceptor, primary electron acceptor, and from there the electrons travel down the redox potential through the cytochromes into PS1. Again in PS1, the chlorophyll A pigment accepts light energy and these electrons get excited, and another electron acceptor accepts this electron and passed on to NADP+ molecule, which reduces into NADPH. We learned that the electrons activated from PS2 are picked up by the electron acceptor, right? Therefore, those electrons need to be replaced. This is achieved by the second step in light reaction, that is the water splitting. There is water splitting complex associated with PS2, which is present on the inner side of the thylakoid membrane. And when electron acceptor captures the electrons, the water splitting complex breaks water into hydrogen ions, oxygen and 2 electrons. These 2 electrons will now move into LHC and the process continues. We saw that 2 electrons from PS1 also get activated due to light, and these electrons are replaced by the electrons from PS2. The last step in light reaction is the formation of ATP and NADPH. We have already learned how NADPH is synthesized, but how about ATP? The formation of ATP from ADP by the addition of inorganic phosphate is called photophosphorylation. The formation of ATP from ADP by the addition of inorganic phosphate, that is photophosphorylation. Photophosphorylation happens in mitochondria and chloroplast inside the cells. There are 2 types of photophosphorylation, cyclic and non-cyclic photophosphorylation. Cyclic photophosphorylation happens in the stromal lamellae of the chloroplast. We learned that the stromal lamellae connects to granum. It occurs under 3 specific conditions. When PS2 is absent in the stroma lamellae, when NADP reductase enzyme is absent, and when the light available is beyond the wavelength of 680 nanometer. As the NADP reductase enzyme is absent, the electrons that get excited from PS1 will go to the electron acceptor but won't pass to NADP+ because NADP reductase enzyme is absent. Rather, they will cycle back into PS1 and electrons are circulated within the photosystem, and photophosphorylation happens due to cyclic flow of electrons. That is why it is known as cyclic photophosphorylation. Here in cyclic photophosphorylation, NADPH is not produced, only ATP is produced. Non-cyclic photophosphorylation happens when PS1 and PS2 are available and work in a series. Both systems are connected through the electron transport chain, and both NADPH and ATP are synthesized as a result of non-cyclic photophosphorylation. In cyclic photophosphorylation, only ATP is synthesized, but in non-cyclic photophosphorylation, both NADPH and ATP are synthesized. Now let's learn how ATP is synthesized in photophosphorylation. This mechanism was explained by chemiosmotic hypothesis put forward by a scientist named Peter Mickel. The synthesis of ATP is due to the development of a proton gradient across the thylakoid membrane. This is the thylakoid membrane, and a proton gradient is developed across the membrane. A proton is a subatomic particle with a positive charge. An excess of positive charge or higher concentration of protons accumulates within the lumen and across the membrane, while outside has a lower quantity of protons. Let's see why this happens. We have learned that when electrons become excited and exit photosystem 2, this is photosystem 2, water molecules split, replenishing the electrons lost by photosystem 2. This process also produces 2 hydrogen ions which remain inside the lumen of thylakoids. When water splits, 2 hydrogen ions remains within the lumen and the electrons move to photosystem 2. The electron acceptor that receives electron from photosystem 2. This is photosystem 2. When electrons get activated, it will reach here. This is the first electron acceptor. This electron acceptor, more than just accept electrons, it also takes up protons. When this acceptor receives 2 electrons, It simultaneously captures one proton from the stroma, facilitating its transport to the cytochrome complex. This is the cytochrome complex, cytochrome B6F. However, the cytochrome complex exclusively accepts electrons, not protons. So what happens to this proton? This proton will released into the lumen inside the inner membrane. Again, there will be an accumulation of protons. Already there are protons from water split, and now again protons are coming from cytochrome. The NADP reductase enzyme is located on the stromal side of the membrane. The electrons from photosystem 1, this is photosystem 1, and the electrons from photosystem 1 are accepted by the electron acceptor, leading to the reduction of NADP to NADPH. This reaction requires proton because this is a reduction reaction, right? So it requires protons which are extracted from the stroma, resulting in decrease in the proton concentration within the stroma. This creates a proton gradient across the thylakoid membrane with fewer protons in the stroma and more in lumen, resulting in lower pH in the lumen. Now this proton gradient or difference in the proton concentration across the membrane of the thylakoid, that will help ATP synthase enzyme. This is ATP synthase enzyme. It is also known as ATPase. This produces ADP from ATP. So this proton gradient helps the enzyme to produce ATP. The ATP synthase enzyme has two parts. One is called CF0. This CF0, which is embedded in the thylakoid membrane and helps to transport protons across the membrane. The other part is CF1, which is protruded outside the membrane into the stroma side. The proton gradient provides energy for CF1 to change its shape, change its conformation, that allow inorganic phosphate to be added to ADP, producing one molecule of ATP. This conformational change of CF1 will also transport protons from the lumen into the stroma through CF0 and ATP synthase. ATP produced is used in the reaction for fixing carbon dioxide and synthesis of sugars. Now we have learned how ATP and NADPH form as the product of light reaction. They are used in the production of sugars. This is called biosynthetic phase of photosynthesis. The production of sugars is known as biosynthetic phase of photosynthesis. That is not light dependent reaction, as it depends upon only the products of light reaction. But this might not be called a complete dark reaction, as immediately after the light is made unavailable, the biosynthetic process continues, but after some time it stops. This is because light is needed for the production of ATP and NADPH, which are necessary to conduct biosynthetic phase. We have learned in previous classes that carbon dioxide combines with water to produce sugars. However, scientists remain uncertain about the mechanisms involved in the process. Just after World War II, Melvin Calvin used radioactive carbon-14 in algal photosynthetic studies and concluded that first product of carbon dioxide fixation is an organic acid with 3 carbon atoms in it. This biosynthetic pathway is known as Calvin cycle. The first product of carbon dioxide fixation is a 3-carbon compound. That 3-carbon compound is 3-phosphoglyceric acid, PGA. Scientists then conducted experiments to see whether all plants have PGA as the first product. These studies revealed that there are some other plants that have a 4-carbon organic acid as the first product of carbon dioxide fixation, which is called oxaloacetic acid, OAA. Hence, there are 2 pathways for Calvin cycle. C3 pathway, in which the first product of carbon dioxide fixation is 3-PGA, and C4 pathway, in which the first product of carbon dioxide fixation is oxaloacetic acid. The first product of carbon dioxide fixation in C3 pathway is PGA. The product is a 3-carbon compound, right? So the molecule that accepts or fixes carbon dioxide will have how many carbon atoms in it? Our answer will be a 2-carbon compound. 2 carbon compounds after receiving one more carbon atom from carbon dioxide will become a 3-carbon compound. But this is not true. After conducting many experiments, scientists proved that the compound that fixes or accepts carbon dioxide has 5 carbon atoms in it. It is a 5-carbon ketone sugar called ribulose biphosphate, RUBP. So the molecule that fixes carbon dioxide is common for both the pathways. It is known as RUBP, ribulose biphosphate, which is a 5-carbon compound. Now let's learn the Calvin cycle. In the Calvin cycle, the carbon dioxide from the atmosphere is converted into carbohydrate or sugars by using the products of light reaction ATP and NADPH. This process occurs independently of light. This means light is not needed for this reaction. The whole Calvin cycle can be divided into 3 stages. First carboxylation, then reduction, and then regeneration. Let's understand one by one. Carboxylation. We have already learned that the molecule that fixes or accepts carbon dioxide is a 5-carbon compound called ribulose biphosphate or RUBP. This RUBP combines with carbon dioxide by the activity of an enzyme called RUBP carboxylase. That is why the reaction is called carboxylation reaction. As a result of this reaction, a 6-carbon compound is formed, but this 6-carbon compound is not stable and immediately breaks into 2 molecules of 3-carbon compound, which is known as 3-phosphoglyceric acid, 3-PGA. The enzyme RUBP carboxylase has an oxygenation activity as well. That means it can combine with carbon dioxide and oxygen. Hence, it is also called RUBP carboxylase oxygenase or Rubisco. But the product of this oxygenation reaction is useless in Calvin cycle. So, as a result of carboxylation reaction, 3-PGA is formed. In reduction reaction, the 3-PGA formed in carboxylation reaction is reduced into a carbohydrate. Here comes the role of ATP and NADPH from light reaction. ATP provides the energy for the reduction reaction and NADPH provides the electrons to reduce or convert 3-PGA into carbohydrate. The carbohydrate formed is G3P or glyceraldehyde 3-phosphate. In this reaction, ATP is converted into ADP and NADPH is turned into NADP+. Both will return to the light reaction to be converted back into ATP and NADPH. One carbon dioxide molecule needs 2 ATP and 2 NADPH for reduction reaction. Now comes the regeneration reaction. In the first step, that is the carboxylation reaction, one molecule of RUBP is used, which has to be regenerated to continue the Calvin cycle, right? So the G3P molecule is produced in the reduction reaction. Some of the G3P molecule is converted into glucose, while the rest is used to regenerate RUBP. Some G3P use 1 ATP from the light reaction to form RUBP, which can be again used to combine with carbon dioxide and the cycle continues. If you observe the Calvin cycle, we can see that 3 molecules of ATP is used, 2 in the reduction reaction and 1 in the regeneration reaction. 2 molecules of NADPH are also used in Calvin cycle. To balance the ATP and NADPH used in Calvin cycle, cyclic photophosphorylation occurs, producing only ATP without NADPH. 6 molecules of carbon dioxide or 6 rounds of Calvin cycles are required to produce 1 molecule of glucose. So how many ATP and NADPH are used? For 1 round, 3 ATP and 2 NADPH. For 6 rounds, 18 ATP and 12 NADPH are used. We already learned that in C3 pathway, the first product of carbon dioxide fixation is 3-PGA, this one. And that of C4 pathway is oxaloacetic acid. Calvin cycle is common to both the pathways. Let's see how. In C3 plants, RUBP carboxylase is present in mesophyll cells and it directly combines RUBP and carbon dioxide to form the first two molecules of 3-PGA. We learned this in carboxylation reaction. This is C3 pathway, but we know that RUBP carboxylase can combine with carbon dioxide and oxygen. Oxygen and carbon dioxide both enter the mesophyll cells and RUBP combine with carbon dioxide and a small amount of oxygen as well. Hence, the carbon dioxide fixation rate is reduced. When RUBP carboxylase oxygenase enzyme combines with oxygen, instead of forming PGA, the product will be one molecule of phosphoglycerate and phosphoglycolate. Neither the synthesis of sugars nor the synthesis of ATP happens here. This process is called photorespiration, which has no value in Calvin cycle. Instead, it will reduce the rate of photosynthesis. To prevent photorespiration in C4 plants, the enzyme RUBP carboxylase is absent from the mesophyll cells, instead located in the bundle sheath cells. This leaf structure is called Kranz anatomy. Kranz refers to a wreath that signifies the organization of cells. Bundle sheath cells form several layers around the vascular cells and are characterized by a large number of chloroplast, and the walls are impervious to gaseous exchange. Hence, oxygen and carbon dioxide will not directly enter the bundle sheath cells. In C4 plants, the RUBP carboxylase oxygenase enzyme is present only in the bundle sheath cells, not in mesophyll cells. Here, when oxygen and carbon dioxide enter the mesophyll cells, carbon dioxide gets converted into HCO3-. This HCO3- now combines with a 3-carbon molecule phosphoenol pyruvate by an enzyme PEP carboxylase. When they combine, the product formed is OAA, oxaloacetic acid. That is the first product of carbon dioxide fixation in C4 plants. Now, malate dehydrogenase enzyme converts oxaloacetic acid into malate, which is another 4-carbon compound. Malate is transported into the bundle sheath cells. This malate is broken down into pyruvate by malic enzyme and carbon dioxide is liberated in this process. This carbon dioxide liberated will then combine with RUBP by Rubisco enzyme and the Calvin cycle continues and glucose is formed. The pyruvate formed is transported into mesophyll cells where it gets converted into PEP and the cycle continues. Like this, C4 plants boost productivity and yield by minimizing photorespiration caused by oxygen. So the C4 pathway is when oxygen and carbon dioxide enter the mesophyll cells, carbon dioxide gets converted into HCO3- and this will combine with phosphoenol pyruvate by the presence of PEP carboxylase enzyme into OAA, oxaloacetic acid. This oxaloacetic acid then acted upon by malate dehydrogenase and converted into malate. This malate is transported into the bundle sheath cells and in the presence of malic enzyme, malate is converted into pyruvate. In this process, carbon dioxide is liberated. Carbon dioxide now combines with RUBP with the presence of Rubisco enzyme and it is converted into sugar. This pyruvate is transported back into mesophyll cells as phosphoenol pyruvate and the cycle continues. Now let's learn the factors affecting photosynthesis. The rate of photosynthesis is very important in determining the yield of plants. It is affected by both internal and external factors. Internal factors are number, size, age and orientation of leaves, mesophyll cells and chloroplast, internal carbon dioxide concentrations and the amount of chlorophyll. External factors are sunlight, temperature, carbon dioxide concentration and water. Here several factors affect the rate of photosynthesis. Hence the Blackman's law of limiting factors is applicable here. The law states that if a chemical process is affected by more than one factor, then its rate will be determined by the factor which is nearer to its minimal value. It is the factor which is directly affects the process if its quantity is changed. That means even if all the factors are optimal, the process will only occur at the rate of available limiting factors. For example, despite the presence of green leaf, optimal light and carbon dioxide conditions, the plants may not carry out photosynthesis if the temperature is very low. Here the temperature is the limiting factor, and if we provide optimal temperature, the process will start. First external factor that affects photosynthesis is light. We know that when light falls on the pigments, the electrons get activated and ATP and NADPH are formed. When more light is there, more electrons will get activated and more energy molecules are formed. More NADPH and more ATP are formed. Then it will reach a point where there is abundance of ATP and NADPH, but not enough carbon dioxide to carry out the Calvin cycle. Hence, if light energy is more, after a saturation point, the rate of photosynthesis will not increase. Here, the limiting factor is carbon dioxide. The high intensity incident light beyond a point causes breakdown of chlorophyll and a decrease in photosynthesis as well. Now let's see how the carbon dioxide concentration affects photosynthesis. Carbon dioxide is present in a very small quantity in the atmosphere and hence it is a limiting factor in photosynthesis. In low intensity light, neither C3 nor C4 plants respond to high concentration of carbon dioxide. If we draw a graph, the rate of photosynthesis in C4 plants is linear at the beginning. Here the graph is linear at the beginning but reach a saturation state where the graph is steady. No more photosynthesis occurs. This is because we know that in C4 plants, the RUBP carboxylase oxygenase enzyme is present in the bundle sheath cells. And when more carbon dioxide is there, more malate gets transported into the bundle sheath cells and more carbon dioxide is liberated because of its reduction. Here, carbon dioxide is higher in the bundle sheath cells and there won't be enough RUBP to combine with carbon dioxide and hence it reaches a saturation point. But in C3 plants, as the RUBP combine with oxygen and carbon dioxide, they need more carbon dioxide to reach the saturation point. The amount of RUBP combines with carbon dioxide is less compared to C4 plants due to the photorespiration. They reach a saturation point after the C4 plant. C4 plant reaches saturation when the atmospheric carbon dioxide concentration is 0.036%. And C3 plants it is 0.45%. Some C3 plants like tomato and bell peppers, which are also greenhouse crops, are allowed to grow in carbon dioxide rich atmosphere to get more yield by increasing the rate of photosynthesis. Now next is temperature. The C4 plants show a higher rate of photosynthesis in increased temperature as the PEP enzyme is more active in high temperature. But in C3 plants, the photorespiration increases in high temperature and hence there will be a decrease in the rate of photosynthesis. Water. If there is less water, it will lead to closure of stomata, and there will be less intake of carbon dioxide. That will decrease the rate of photosynthesis. Moreover, water stress leads to the wilting of leaves, and that will reduce the surface area for absorption. That's it about the chapter photosynthesis in higher plants. If you like this video, please share it with your friends. If you have any queries, put it in the comment box. Thank you.